everyone, and I'd like to take this opportunity to thank Rajiv for giving us this chance to tell you a bit about our project, and also I thank Jun Wang for introducing it to you. And it's a collaboration between BGI Shenzhen, the Chinese Academy of Agricultural Sciences, and IRRI. So to start off with, I'd like to give credit to the different teams, the group at BGI that did the heavy lifting, the preparation of the libraries and the sequencing and the bioinformatics that's been done so far, our group at IRRI and at CAS, teams that have been involved in the purification of the plant material and the increase of seed, and now working with data, as well as the donors, Bill and Melinda Gates Foundation for funding to the Green Super Rice Phase 2 project, Chinese Ministry of Science and Technology, and initially also it's a collaboration with BGI, and BGI has supported this extensively. And both IRRI and CAS have had other sources of funding that have contributed to the preparation of the material going into the sequencing. And since we are now putting all of the purified lines into our gene bank as new accessions, the Global Crop Diversity Trust is providing support for that activity. So a brief overview. I'd like to introduce our research organization under the Global Rice Science Partnership, and what we're doing to build a genetic diversity platform for rice, an introduction to our gene bank and the Chinese National Crop Gene Bank rice collections, how we undertook selection of material for the sequencing effort, a bit about our now 3,000 rice genome sequences, and what we are doing in preparing for high throughput phenomics, as well as how to adapt to very large data sets under our newly established International Rice Informatics Consortium. So under GRISP, or the Global Rice Science Partnership, this is a program under the reorganized CG system. It includes our work at IRRI and Sister Center Africa Rice, but also the rice program at CIAT and non-CG centers, predominantly CIRRAD and IRD in France and JURCAS in Japan, but now many other collaborating institutes and partners as well. And under that research program, we have 6 different themes of research, and the first theme is everything to do about germplasm characterization, utilization, and creation of new genetic stocks. And so we have the gene bank, conservation, dissemination, 1.2, which is characterization by molecular and other means of genetic diversity, and creating new populations for use in genetic studies. So we have a rice SNP consortium where we've undertaken the genotyping of about 2,000 purified accessions with 700,000 SNPs. That work is now being readied for publications, collaboration with Cornell, Susan McCuch's group. We have a global phenotyping network established that's undertaking phenotyping for a wide range of traits, starting with diversity panels coming from the 2K set of material that has been genotyped on the high-density rice array, but also now feeding in material from our resequencing effort. And then this project is under the whole genome resequencing of gene bank stocks. Then we also have several projects undertaking the creation of new specialized populations, including about 12,000 RILs from using parents that were used in a previous SNP discovery project, several different MAGIC-type populations, both within sativa as well as using wild AA genome accessions as founders, and some other specialized population creation, but also our targeted gene discovery for high-value QTLs and the C4 rice project. All of this is feeding into having a public genetic diversity research platform with genetic resources as a foundation, wherein through combining genotype and phenotype data, we can get at the holy grail, gene function, and bridge the genotype to phenotype divide, and be able to address these challenges that are of high importance for breeding efforts going on now, including drought tolerance, grain quality, durable resistance, and other abiotic stresses. And by adding this degree of information into the gene bank collection, will improve use of the collection by also having the increased level of information attached to the material, and justify the extent, to some degree, the extent of conservation of material. All of that, though, requires that we have access to large-scale computation, bioinformatics, and databases to be able to deal with the data. So in terms of germplasm feeding into the effort, our institute has the International Rice Germplasm Collection, and we have now about a little over 120,000 registered and incoming accessions from Asian cultivated rice as well as African cultivated rice, and affiliated species within the genus Oryza and some affiliated genera. But to date, very few of those accessions have fed in donor alleles into modern high-yielding varieties. So is there some process that we can undertake to identify new sources and feed them into breeding programs? And that's our ultimate intent. For the China National Crop Gene Bank, they have about 72,000 accessions of rice in their collection, both cultivated, wild, and I guess these are genetic stocks, and I'm not sure what they meant by sea rice. This was information gleaned from their website. And Li Zhikao undertook the development of a core collection from their rice collection, largely using morphological characterization data, and that their mini core collection fed into this project, as well as material coming from the working collection for the Green Super Rice project. So historically, starting in the mid-'80s until the late 1990s, about 23,000 accessions in the IRRI collection were characterized by isozyme analysis. And to date, this is the largest collection of molecular data that we have on hand for rice. It's largely, the material was largely geared to Southeast Asia in terms of the total number. But if you take globally across all of these different partitions, then what we see is that about 3/4 of the material, if this is indeed a random sample, 3/4 of the genetic diversity of rice is within the indica subgroup. So, and about 1/4 tropical japonica, the other groups are relatively minor. So if you can keep this in mind for a little later. So in terms of germplasm selection, when I joined IRRI's Gene Bank in 2001, I set out to design a core collection. Not a core collection from the standpoint of reducing the management of our gene bank, but a core to have a representative sample of material in the gene bank. And to do that, genome type, subspecies if it was identified, source country in terms of location, whether or not there was a indication that the material had been used in at least IRRI crosses by tracing pedigrees in the IRIS database was factored in. But there was a considerable degree of random selection, and that was done in a way to try to evenly cover the namespace. So materials that had similar names were preferentially selected at a lower rate, unless in the instance like we have 8,000 accessions from Laos, and many accessions just have the name cow or kam, meaning rice. So those were sampled at a lower rate than 10%, but still at a sufficient rate to try to get good coverage of that. And then added to this led to about 12,000 accessions being selected from the collection. And then over time, because of various project activities and so forth, we've added more material to the set, so that we've now been working with about 15,000 accessions in terms of purification and having seed stocks ready to go for additional sequencing and characterization in the future. So for our set of 3,000 materials, about 2,500 came from this core collection. That was after we took into account the material being used under the high-density rice SNP genotyping project. And then the rest were essentially a random selection from the remaining entries in that 12K core. But we also included nominated materials coming from projects led by Sirad, notably a panel that was termed the Oritage panel, and then specific donor lines, for example, those materials that were used as founders for our Indica and Japonica magic populations. Then coming from the Chinese National Crop Gene Bank and GSR working collections, there are about a little over 500 entries that fed into the sequencing effort. All of these have undergone at least one round of single seed descent purification prior to getting the plant that was used for isolating DNA and growing to maturity, getting the first generation of seed, and then that seed flowing into further seed increase. So at this time, we have about 2,200 plus of these where we now have new accession numbers assigned. The other materials are in the pipeline for assigning new accession numbers, and we need to obtain from CAS the material used from China. So in terms of the global distribution of the genotypes, majority are coming from a little over about 34% are from Southeast Asia, 26% from Southeast Asia, and about half of those are, for the sake of the audience here, are Indian accessions. A large fraction of the rest are accessions from Bangladesh, where there are some minor variety groups that are predominant in that region. And the rest globally distributed, and in parentheses, the number of countries represented. It's a little over 90 countries that have been sampled for this effort. In terms of the sequencing, sequencing was done by BGI using Illumina-based platform, single library, so this is resequencing data. And it's mentioned we have sequencing data now on 3,000, 17 terabases of filtered trimmed reads that have been produced with an average depth of about 14x per genome. And the range was from a little bit less than 5x to even over 60x in a couple of cases. BGI undertook initial informatics analysis, predominantly the use of BWA and the GATK pipeline with variant calling relative to the IRGSP 1.0 build of Nipponbari that was released last year, the unified build. And we now have additional analyses underway with a large consortium of partners looking at population genetics and trying to build de novo genomes and other work. So the data, it's uploaded into GigaDB. Each of the sequenced accessions is getting a data object identifier, a DOI assignment. We've been using in-house Galaxy documentation, and we're suggesting this as a mechanism under a consortium for allowing us to track and document workflows and analyses. We've submitted now a data note and commentary paper. The data note went to GigaScience Journal at the end of January 2014, and then 2 weeks of Chinese New Year and Chinese Spring Festival has intervened. We are anxious to know how quickly this is going to come out. But this data note releases the FASTQ data files, the accession list, and provides information on how to get seed, and the commentary paper indicates our path to further data analysis. And several times at various venues, we've called on additional partners to come in and help us with more value-added analyses so that we can bring this data set into a form that will be useful for application to breeding. So just some results from data quality assessment that was done by Nikolai Alexandrov, a recent addition to our team at IRRI. And this was from 1,000 genome sample, and the degree of failure rate was reasonably low. And one thing that stood out was the per base sequence quality of about 5%. On looking at this data further, Nick took some of the reads that were in the poor sequence quality and also noted that there's a bimodal distribution in the reads. And it turns out that those reads that were in the second peak, when they mapped, when they were blasted against the NCBI database, then what came up were hits to Xanthomonas. So some of the material that was, plant material that was used for the sequencing had a low level of infection with pathogen. So there is a way that we can, instead of calling it a failure, we may have gotten additional information about the biome of rice in terms of bacterial and maybe fungal organisms. So Tai Shuai at BGI, then 200,000 random SNPs, 5 sets of 200,000 random SNPs were selected. TreeBest was used to calculate maximum likelihood distance. Matrices with 1,000 bootstrap replicates on each of those 5 independent sets of random SNPs. And then consensus trees for those sets were created, and then a combined consensus tree across that was created, and then consistency of group placement for each of the 5 variety groups was assessed. And when you see the color superimposed on the topology, then a strict consensus, simple majority rule consensus placement to that group occurred. So the largest fraction of accessions, about half, roughly 1,600, fell into the indica group. And then we have a rather smaller group of Alsporo intermediate types, the aromatic basmati sadri types, material that is not easily categorized as either temperate or tropical japonica. And then within tropical japonica, 2 distinct clades, and then temperate japonica. and perhaps on the order of 5 separate clades within the indica. Based on that classification, then about 60% were indica. And if you see this based on SNP, classification in the groups relative to the isozyme variety group distribution, it's relatively similar. So even though this was not perhaps the most optimally designed set of material to encompass a core set for rice, it seems that we have quite reasonable coverage. Just some basic information about the SNPs. SNPs were then mapped using the MSU gene models and characterized. And then one thing about the SNP set, it's about 19 million SNPs, is that it was a population-based emission of SNPs from the GATK pipeline, which required that minor allele frequency be greater than 0.1%, which translates to a minimum of 3 accessions needed to carry that variant before the variant made it into our data set. Moving beyond our primary analysis, we've begun forming an international rice informatics consortium. We have engaged some advisors for the consortium, and we have now a large team of partners interested in working with us on further data analysis, including iPlant Collaborative, Arizona Genomics Institute, Rod Wing, A-STAR in Singapore, University of Southern California. Some of these partners have come in because of Nick's professional linkages, but also teams that are more familiar to the rice community and have been teams that have worked on the Panbari sequence. So in terms of phenomics, at IRRI, we now have what we call a Pati scanner. It's a GPS-guided tractor with a boom that's the length of one of our Pati areas. And on that boom, there are 8 sensor positions where different sensors are mounted that allow us to collect different spectra regions, infrared canopy temperature, ultrasonic canopy height, different measurements for the local climate, and then other information that's collected that's needed to get down to the plot level. And also a high definition video, just so that you can monitor through the field at this point. And it's allowing us to get estimates of biomass, canopy height, canopy temperature, et cetera. This just shows fields of view from sensor position looking down across the plot. So we have microplots in a design in the field that allow us to collect these sensor streams. So at this time, the system is being used for screening RIL populations and a diversity panel from the 2K set, but this season is our second season of using the system. We're continuing to look at RIL population and indica diversity panel that was not in the sequencing project. Next season, we will be putting in material from our resequenced lines into this field-based phenomics. We also have now a tray-based imaging system with the C4 group that allows 3D image reconstruction of plant architecture. So this is upcoming. I'll just, this is from the RIL population, the near-infrared measurements. It's well correlated with manual measurement of biomass, and it allows you to get at some of the underlying traits with a reasonably good correlation between the manual measurement and the derived measurement. So even though the heritability for these automated measurements is lower, there is the nice correlation between them that should mean that this is an acceptable proxy for the very expensive and laborious manual measurements. So just briefly, our International Rice Informatics Consortium, or IRRI. It was initiated a couple years ago at PAG. We've had 2 meetings at PAG, and we'll have an upcoming meeting again at PAG Asia this May, including a formal workshop on the program. And contact organizations, it's more than 50 companies, universities, foundations, different bodies like ICAR in India and RDA in France, and et cetera, that have expressed some degree of interest in the consortium. So we want to build on existing efforts. There are a number of activities going on in the international community, such as the International Arabidopsis Informatics Consortium, that we can draw on for this. There's an activity now in maize and soybean and wheat, so forth. So the whole crop community is moving in this direction. We do need to have some agreed on standards and ways so that we can integrate data across different databases, as well as we are wanting to have a unique database for our activities that will make use of modern web technology for that, where germplasm is a key focal entry point. And then content would be information from the 3K project, from our 2K project, phenotyping effort, as well as data in the public domain. But we're not trying to duplicate Grameen or other well-known databases for rice in this regard. So our team working on this at IRRI, and in brief, some conclusions. For rice, there are well over 30 million SNPs that have been discovered, both under our project and other projects in Alpha Rice. We've been generating huge data sets of genotyping, sequencing, phenotyping data, but how we integrate and make use of that data is our key limitation and bottleneck. So that led to our formulation of the International Rice Informatics Consortium, so that we can integrate data across sources and facilitate collaboration. All of that hopefully will allow the ability to predict phenotype from genotype so that we can accelerate breeding. So with that, questions?
Thank you, Kenneth. Another great example of international consortium by a CGIAR center to sequence, phenotype, and of course I was very much fascinated by the robotic work to phenotype some of the most complex traits. With that, I would like to take a couple of questions, please.
One last comment is, for those of you who have visited IRRI, our gene bank is right there. So. Cost. Am I at Gunyong? Can I say? It's something over 3 million for sequencing, informatics, and then backstopping.
One question. Yes, here.
I was curious about the bacterial DNA that you have, sequences that you have encountered. There is a bacterium called Stenotrophomonas that's very closely related to Xanthomonas, but it's an endophyte of rice. So I was wondering whether you're actually sampling material that have bacterial endophytes rather than low level of infection with Xanthomonas.
Maybe, but Nikolai did that analysis, and just from the short read, there was quite a good match to one of those Xanthomonas oryzae published sequences. So that's where that bit of information came from. But we're not excluding any other possibility, definitely. Thank you.
Okay. Thank you. I think please join me in thanking Ken and his colleagues for the wonderful work. Now I would like to invite my co-chair, Philip, to give some key messages from the session.
Okay, thanks a lot. Yeah, a few words. The first would be probably linked to excellence in partnership, excellence in science. I think that the last presentation and all the presentations this afternoon were very clear in that sense. Basically, IRRI is collaborating with other centers, Africa Rice, SIAT, with CIRRAD, IRRI, and really the results they are providing are just excellent results for breeding. For CIMIT, collaboration with Cornell University, collaboration with different advanced research institutes in Australia. For ICRISAT, again, collaboration with other advanced research institute in Chickpea. So basically, that's a key message from the CGIAR, our open mind and need to collaborate with different advanced research institutes. So second point, technologies are there. For sequencing, next generation sequencing, resequencing, genotyping by sequencing, so we are fully able to provide a huge amount of data. But the question is, data for what? And we will talk about that in a few seconds. Next point is germplasm. Germplasm is a keyword, and you know that by 11 CGIAR centers around the world, we are taking care of 700,000 accessions and collaborating with other national gene banks. That's providing genetic diversity for GWAS analysis. That's providing genetic diversity for the obtention of recombinant inbred lines, introgression lines, magic populations, and so on. So the pre-breeding material is also available. For phenotyping, we have different technologies and methods for precision phenotyping, and the last example was just amazing. So what's missing for reaching excellence in development? So that's the question. Some few colleagues will say, oh, we need money. That's the problem. Yeah, but money for what? I would say the first point would be money for capacity building. That was highlighted by Peter. We really need very young and well-skilled scientists to be able to bridge the gap between all this data, the tool for using this data, and using this data for breeding programs. Because basically, we need these young, well-trained people to bridge the gap between the lab, between bioinformatics, between the different mapping approach to really provide material for the breeding and for the breeders in the fields. Next point would be linked so to capacity building, capacity strengthening. Then I would have a look to the private sector. I'm coming from the private sector for seed companies, and basically I would say that if we want to have impact in the development of new varieties, we have to clearly separate what's trait discovery and the trait discovery pipeline from the variety, new improved variety delivered to the farmers. These are two different jobs. So we have to in the CGIAR research program, we have to clearly separate both. And then finally, I would say that it's the right time. It's the right time because the 16 CGIAR research program are reaching, I would say, the end of their life cycle. Some of them will end this summer, other next year, but basically what all the CGIAR research program will have to do is to prepare the second call starting in 2017. So it's time to increase the quality of the partnership. It's time to increase capacity building. It's time to think about the integrated breeding platform and next step for this integrated breeding platform supporting breeders' activity. It's time to collaborate with the Center of Excellence in Genomics at ICRISAT. It's time to develop all this kind of new strategy to have a very competitive vision regarding the next program of the CGIAR. So basically, we are thinking that it's the only solution if we really want to have impact, if we really want to address food security, to increase nutrition and health, to reduce poverty, and all these issues in a very sustainable way. So thanks for your attention and have a nice dinner.